Hello everyone, have you ever heard about genome sequencing? Now I am going to share with you the latest finding published in 2017 about this genome sequencing by BMC Genomics. Title of the journal is A Universal Genome Sequencing Method for Rotavirus A from Human Fecal Samples, which identifies segment reassortment and multi-genotype misinfection. So we go to the overview of this journal. The objective is to understand the phylogenetic of rotavirus by using sequencing method, while the problem statement is genomic characterization of rotavirus not being adopted at large scale due to the complexity of obtaining sequences for all 11 segments, particularly when faces are used as starting material. Basic background of rotavirus are it causes diarrhea, it commonly attacks children below 5 years old and mostly happen in poor country. It causes disability, adjusted life year or dailies. These are all the symptoms once affected by rotavirus. This is the structure of rotavirus and this is their characteristic. Basically, rotavirus is a non-enveloped virus and it has 11 segments of protein. The most dominant are VP4 and VP7. Before we go to the methodology, we need to purify the sample. Why? Because fecal has many contaminants that we need to eliminate, such as bacteria, mineral, dirt, and so on. It also contains inhibitors that might disturb the method, and yield of rotavirus that we need might low. There are several methods that perform in this research. The first method is clinical specimens and detection of rotavirus. The second one is the need to prepare the rotavirus DNA for genome sequence. So here, the important step is doing EIA plate to prepare ROV genome. Then, we pursue the genome sequencing by using various kits and technologies to purify the DNA. Illumina also we use to sequence the sample. Next, we analyze the sequencing of rotavirus genome using the software. And the last method is to analyze the phylogenetic tree by assorting the genotypes of each segment. Several results have been concluded from the experiment done. One of them is ROV purification of genome sequences is dependent from fecal sample and viral load. Table 1 shows the concentration of ROV required for genome sequence generation. As we can see here, we perform different rate of serial dilution. The higher the dilution, the higher the CT value. CT value or cycle threshold is defined as the number of cycles required for the fluorescent signal in real-time PCR to cross the threshold. It basically inversely proportional to the target copies of the genome. For instance, by doing the random amplification, as the CT value is higher, the target copies will become lower. Same as ROV genome coverage we got, the percentage will become lower. Thus, we can conclude the best CT value is 15, as we can achieve genome coverage near 100%. Next, validation of method for the genome sequence of different ROV genotypes. From the table 2, by doing the step of the capture and amplification process of several labeled samples, the most dominant genotype that we tested is G1P8 with certain CT value, and here the expected target copies we got. Other than that, ROV genome we tested are G2P4, G26P19, G1P4, G9P4, and G3P4. UT is the unknown. Then, optimization of ROV cDNA amplification for genome sequences. Table 5 shows the sequence coverage of ROV genomes using viral capture and amplification with a random and specific primers. Here, Table 4 displays the primers that use specifically for amplification process based on label sample with respectively genotypes. We got certain values of genome coverage and segment length and coverage in percentage. From here shows the length segment of ROV protein is shortened. Every 100% means the coverage of targeted ROV genome that being aligned and read by the sequencing analysis is the most accurate. Last but not least, we are able to build the phylogenetic tree. Figure 4 describes the genome constellations that suggest intergenotype reassortment in Vietnamese G1P8 and G2P4 ROV. A maximum likelihood phylogeny of PP4 gene sequences is shown with a genome constellation map at the tips of the tree. Standard WA-like genome segments are indicated by light gray blocks, DS1-like segment by dark gray block. Taxa names in the bold indicate Vietnamese sequences from this study. Taxa names in red indicate likely reassortment Vietnamese sequences. From the result, we discussed that the research of the genome had a 100% success rate, where most of the sample they got more than 90% of the genome coverage with a less CT value. This means that genome coverage are successfully covered all 11 segments with different length of segments. Overall phylogenetic tree could be constructed. The conclusion that we got is this method provides a novel, reproducible, and comparatively simple approach of genomic ROV characterization and could be sealed out for the use of global ROV surveillance system.